In this exercise, we are going to use multiple sequence alignment tools to investigate the relationships between insulin proteins in different organisms. For doing this, we are to obtain the sequences to compare from a blast search. Let's go to the EBI website and suppose that you want to include the human insulin protein in the comparison. So we search for the human insulin in the protein database and we obtain a group of hits related to the insulin protein of Homo sapiens. We select one of them, and if we want to be sure that we are working with a good data, we could take the ref-seq corresponding to this entry. Reference sequence are well annotated data of sequences that is included in the NCBI database. Here we have 4 isoforms of the human insulin. We select one of them and in the protein entry we can access directly to a blast search. Here we have the accession number of the protein sequences in the database. We're going to select reference proteins to search for, and in this case, as we want to compare the insulin sequences in different organisms, we can choose here which organism we're going to use for the comparison. For example, the chimp the dog, here in the organism name you can use common name or taxonomic name. For the mouse, the cat, the chicken, and the lizard. These are the animals that we want to investigate the relationship of insulin molecules. We're going to use a standard blast P search, and we can customize and reduce the max sequences, target sequences to obtain in the output, because we're only going to work with a reduced set of sequences. We submit the blast calculation, and here are the results. As you can see, the first hits correspond to insulin molecules, different organisms with good E values, lower E values, that indicates a strong relationship between the human sequences and all these sequences, and also a good query coverage that guarantee that the sequences that you are found are of the same length of the query investigated. Also, we have the alignments, and we can select here the sequence that we want to process. In this case, we don't select the first of chimp sequence because it's annotated as predicted, so it's better to work with non-predicted sequence. We select from chimp the insulin from the dog, the insulin for the mouse, cat, chicken, and lastly the lizard insulin. Once that we have selected the sequences that are of the organisms that had a good hit with the human sequence, we can download as a FASTA file and here we are, the collection of sequences that we want to compare. Here, it's better to modify the first line of the FASTA file sequences in order to facilitate the further analysis of the results. So here we can reduce the name of all sequences and we keep the name of the species because we are pretty sure that all these sequences correspond to insulin molecules. Well, in that way, we change the names of all the sequences and include the human for the comparison. Now, the next step is going to a multiple sequence alignment program. We're going to use the European Bioinformatics Institute custom tool. You have to remember that in the European Bioinformatics Institute there are multiple tools for doing such alignments. You can choose from one of these tools if you want to build a multiple sequence alignment. We're going to use Crucial W, but you can use any of these softwares. So let's go to enter the sequences to be compared. You can change parameters like the substitution matrix and the gap opening and gap extension penalties, and also other parameters related to the algorithm. In this case, we're going to maintain the default parameters and we're going to submit the alignment. Here is our result. As you can see, all the sequences are aligned and according to the legend on top on the bottom, we can see that there are regions of high similarity between all sequences. We can see this more clear if we display the alignment in a color code according to the type of amino acid. We have here the result summary of our alignment. When you can see the resulting pairwise comparison of all sequences in the line. You can organize by the score of the pairwise alignments. As you can see, the highest score alignment corresponds to the comparison between the human and the chimp sequences, and the worst score corresponds to the alignment of the lizard and the mouse sequences. You can consult also the guide tree that shows the connections between the sequences according to the distance matrix calculated using the scores of the pairwise alignments. Here you can see that chimp and human are close, same from dog and cat, and the rest of sequences. You can modify this view to adjust the distances to a more biological significant view. We see that the vertebrate sequences or mammal sequences are more closely than the chicken or lizard sequences. This is more explicitly easy to see if you use an application as the JALView application of the alignment of the sequences. Here we have the alignments of our sequences. There are some program JALView give some graphical tools for obtain for easily interpret the alignment. Here are a region of strong conservation. Here are regions of less conservation. We can color the alignment as by identity percentage, and it's very clear from this color view which are the regions that are very conserved between all insulin molecules between the species, and which are the regions more or less conserved. And also can calculate some tree that exemplify the relation between all the sequences compared. We see that the human and the chimp are very close, unless, and then all the mammals, insulins, are related, and more distance we found chicken and the lizard insulins molecules. This is a simple way to use the NCBI web page for doing a blast search, collect a group of related sequences, and analyze them using a multiple sequence alignment